my name is Mark Bennett, and I'm a researcher at the Walter Eliza Institute, and I'm also based part-time at the University of Melbourne. And I work with the epilepsy team led by Professors Ingrid Scheffer and Sam Berkovic. And you're going to hear from Amy, also part of the team after me. And so I'm a bioinformatician, so I'll tell you what that means in a minute, but I'm going to talk to you about what bioinformatics is and some of the approaches that we use to try and identify and understand causes of genetic epilepsies. So what is bioinformatics? I think every single talk I've ever been to, when they explain what is bioinformatics, they use this exact picture, but there's a good reason because it really captures it quite nicely. So it's kind of it's a sort of multi-disciplinary area, and it's taking skills from biology, from computer science, and from maths and stats, and sort of putting them all together. And to try and put it into words, bioinformatics is using computers to analyze and understand large, complex biological data sets. So the modern advances that we have in medical research and genetic sequencing, we get these incredibly rich, amazing data sets, and being able to harness those data sets and derive insights and understand them, that's where bioinformatics comes in. It's a sort of a new field that's come up in the last couple of decades. So in this talk, I'm going to try and answer some of these questions. So how do we identify and diagnose genetic changes? And so Emma gave a great talk yesterday, and this is sort of, I guess, going to give you a little bit of the nuts and bolts behind, if you do genetic testing and you get a result, what actually has happened to identify those variants. How can we try and understand the effects or the function of particular genes or particular genetic changes? And one question of many that we're interested in is why do you see people that have the same gene or the same individual genetic change, but they can have, in some cases, very different types of epilepsy? And how can we look at genetics to try and understand what is the cause of this? So just to start really simply, DNA, we have these 4 letters, A, C, G, and T, that make up the 4 nucleotides that form DNA. And so these letters get put together to form your genome. So your genome is your own personal DNA sequence. And we have about 3 billion of these letters, together in some particular configuration, and that's what defines your particular genome. So to give you some context about what exactly 3 billion letters looks like, if you count all of the individual letters in The Hobbit and The Lord of the Rings, the complete works of William Shakespeare, Tolstoy's War and Peace, you haven't even got close. So you basically get a stack of books roughly the size of the Sydney Opera House to get 3 billion letters. So this is incredibly complex, deep amount of information that we're trying to process and understand. And essentially, when we're looking for trying to diagnose the particular genetic change that is the cause of epilepsy, essentially it's like trying to look through this almost small library of books and trying to find one single typo or spelling mistake. So how do we do that? DNA sequencing has just advanced massively. So it's incredibly powerful, and the advances in sequencing technology that have come in the last decades and continue to come and are going to come in the next decade are just mind-blowing to me. The first human genome was sequenced in 2001, and it cost $3 billion and took 13 years. And now we can do that, and it costs a couple of thousand dollars to do. To sequence DNA, you get someone's DNA, you chop it up into little pieces, you feed it into a sequencing machine, and this reads along the DNA, and it gives you these strings of DNA that look something like this. So each individual little piece of DNA that goes in the machine comes out with a string of DNA letters. And this is the raw data that comes to myself or to bioinformaticians, and then we're trying to process this data, analyze it, and make sense of it. And we're working, again, the numbers here are kind of mind-blowing. You have hundreds of millions of these pieces of DNA in a single sequencing experiment. So bioinformatic analysis is how we process this data and try and understand it and derive meaning. And so typically what we do is we take these individual pieces of DNA and we try and put them back into the right order and find where they come from in the human genome, where in those 3 billion letters they come from. So if you think back to my picture about this enormous pile of books, imagine you put it through a shredder and you're trying to put it back into order. So to do this, we use some very powerful computers and computing algorithms. And essentially we take these individual pieces of DNA, these DNA sequences, we match them back up to what's called the reference genome. This is kind of like the standard human that everyone gets compared to. And by doing that, we end up with a picture that looks something like this. We have all these different pieces of DNA that have been sequenced, and we can go along, scan along the genome and look for where is there a change that here we have a G that's changed to a C. So this is one particular genetic change. We call these genetic variants. So the second challenge, I guess, is that if you take any person and you look at your own genome, each one of us has something like 3 million genetic variants. Majority of these, or many of these, are common, common across many different people. They might explain why someone's taller or shorter than someone else, why your hair is a color, your eye color, etc. But even still, most of us will have approximately 100, I shouldn't say unique, approximately 100 variants, which you didn't inherit from either your mother or your father. They just occurred due to spontaneous changes in DNA. So the amazing power of modern DNA sequencing technology is we get this incredibly rich data set. We can essentially sequence your entire genome, find out almost all the genetic information that explains what makes you you. But the challenge is that we have this incredibly rich data set, and how do we even make sense of so much information? So to do that, to go through 3 million genetic variants would be impossible for a human. Even to go through 100 would take a lot of time. So this is where, with bioinformatics, we use a variety of different computer algorithms to try and process this data, make sense of it, and come up with interesting results. And people sometimes talk about this as trying to sip from a fire hose. You have this absolute torrent of data that we're trying to manage. So hopefully you can see some of the challenges that we're facing, and hopefully I can tell you a little bit about how we try and do this. So one technique that's very powerful is machine learning. You may hear people talk about machine learning, artificial intelligence, deep learning, different varieties, and there's a few different varieties, but essentially it's a method where we try to teach a computer how to train the computer so that it can then do the job for us. So to give an example, a simple example is how this works. Let's say we want to train a computer to recognize handwriting, in this case, just numbers. So we take some sort of training set, we give the computer a lot of data, and we say, these are the zeros, these are the ones, etc. That goes into training data. There's many different machine learning algorithms. These are generally complex mathematical models about that you can take the algorithm, take the training data, and the computer will kind of optimize and come up with a particular model. And then once we have our model, we can take some other data, some test data. Here we've got a different set of handwriting, and we want to see, does this work very well? Each one of these things goes into our model. The computer's trained what it thinks, will help it recognize a 4, for example, and we're going to get a prediction, and does it actually work? So we test the model. If it doesn't work, we go back to the drawing board. Maybe we look at a different machine learning technique. But generally, with a bit of work, we can come up with a good model. And now we can take this model and use a computer to look and try and accurately analyze and classify, hundreds, thousands, millions of genetic variants. So this is a simple example, but we do use machine learning to train models for how to identify, the genetic variants. So we can look at different features that genetic variants might have. We can feed training data in because, huge amounts of research and studies which have looked at different genetic variants. We can take that and try and train a computer to be able to do this at the massive scale that we get from the genome sequencing data. So interpreting genetic variation, this is the challenge. And this is what we, hopefully we can, look at all these variants and narrow down to what is the single variant that is causing disease. And as I said, using computers to really try and make this efficient and focused. So this is one example. This image here is based on a study, and they looked at all of the different voltage-gated sodium channel genes. So there's 10, I think, different sodium channel genes there. Some of them you might recognize that cause epilepsy, and some of them don't, or aren't yet known to be associated with epilepsy. But all these genes come from the same family, so they have some shared common features amongst them. And by combining data across all these genes, the hope is that you can get more information that might help you interpret any single one. And using some of the incredibly large public databases that exist, which really make our job possible. The idea here is to look across the sodium channel genes, the different domains, different regions in these genes, and identify which regions are most likely to be at risk for a particular variant that is going to cause disease versus something that won't cause disease. So the light teal pattern in the background, this is sort of the pattern of variation you see in very large databases of hundreds of thousands of people that we think are unaffected, so not known to have disease. And then the purple here is the opposite. So these are variants that have been identified and believed to cause disease. And so if you look across here, you can see that in some places, there's many of these teal variants. These are neutral variants that don't cause disease, and very few variants that do cause disease. And in other places, it's the other way around. And so this suggests to us certain regions of these genes actually you know, if you have a change there, it's probably okay. That region of genes maybe not critical, whereas these other regions, you know, which do have a lot of disease-causing variants, are probably very critical regions that are important for that gene's function. And so on the bottom, you can see there's these little red spikes, and they in the study they define 16. They call them pathogenic enriched regions, but these are 16 little regions where any variant in there, you'd be more likely to cause disease than to not cause disease. And so by taking methods like this and many other different approaches, combining it all together, we try to take, another method, looking at protein structure. So this is an example for a gene CHD2. So the gene code in terms of the DNA is I've sort of trimmed it down to be short, but it's, you know, it's thousands of base pairs. But this gene gets converted to a protein, the protein folds up, and you get this actual 3D structure. And so looking at DNA as a string of letters is very different to looking at what in reality the actual 3D structure of the protein is. And so again, there's very powerful machine learning models that have been developed, which now allow us to predict the structure with incredibly good accuracy for any protein at all. And the results are, especially in that have come in the last about 12 months, have been astonishing. But looking at the protein structure gives you a very different picture of what is going on in this particular gene than looking at just a series of letters, because you can see the protein folds around and twists on itself. And so two things that might be very far away in the DNA, in the genome, actually it's sort of a close because of this twisting. And so you can get new and different information by combining all these different things. And so by doing this, taking all this information and putting it together, we try to classify the different genetic variants. And typically, if you've had genetic sequencing and had a report, you might have got a report that, you know, that a particular genetic variant was identified that's it's classified as pathogenic or classified as likely pathogenic. Or it's a variant of uncertain significance. And Emma spoke about this yesterday. And so if you've had this genetic sequencing, I guess, I hope this gives you a little bit of an insight into like what's going on behind the scenes that you may not have been aware of, and sort of where we're still working to try and improve things. So many variants of uncertain significance, hopefully as time goes on and as knowledge increases and as the methods and sequencing and technology gets better, we'll be able to reclassify these variants and say either whether they're pathogenic or whether they're benign. And so currently about half the time you get a genetic diagnosis, but still half the time you don't. And so in those cases, how are we thinking about trying to identify genetic diagnosis for people who don't get a diagnosis through this analysis? I think Steve showed this image yesterday about looking at different types of genes that are in different groups. And so one way you can start is you could look at, for example, ion channels. And as I said before, there's actually 10 different sodium channel genes, and only 4 here that have been linked with epilepsy so far. So maybe other ones are places to start. Or the mTOR pathway on the bottom left here. mTOR pathway is known to play an important role. You know, a number of different genes that regulate this pathway have been linked with epilepsy, but maybe many other genes in this pathway also play a role which hasn't yet been realized. So by starting with some of these genes that are not yet known to be associated with disease, but are related with other genes that are associated with disease, we can start to try and expand our knowledge. The other place that we can look for people that don't have a diagnosis is the missing 98% of the genome. So most genetic sequencing analysis typically focuses just on the coding regions of genes. So these are the 2% of the genome that actually gets converted and ends up as a protein. There's a very large amount of the iceberg below the water that in many cases we don't look at. And this image, unfortunately, is a little bit blurry, but hopefully you can still get an idea. Identifying these genetic variants through sequencing we can do that, that's fine. Understanding them is the real challenge. And so understanding variants that sit outside of these coding regions is a really interesting and important challenge, because many of the people who don't yet have a genetic diagnosis, likely the answer is going to be sitting in this missing 98% of the genome that's not really being looked at closely. And so there's a number of different people trying different approaches using machine learning and other techniques to try and take information that we have and look at the non-coding part of the genome and try and understand where genetic variants that might, so they won't directly affect the protein, but they might affect the regulation. So, you know, gene might be not controlled in an appropriate manner, and that can still lead to disease. Okay, so finally, I want to talk about a little bit about phenotypic variability. And this idea, I think, was mentioned a couple of times yesterday, but the idea that, you know, you have a number of different people. So we have 4 different people here, and they all have perhaps mutations in the same gene, or they might all have the exact same genetic variant, some sort of, they're related through some sort of family tree. And I'm representing this here by this little box. So everyone usually gets 2 copies of a gene, one from your mother and one from your father. In this case, the one in orange is the gene that has the particular variant. So we might expect that everyone with the same gene or with the same exact genetic change would have the same type of epilepsy. But in reality, we see many examples where this is not true. And so even people, that have, like even siblings have the same genetic variant actually have quite different types of epilepsy. And so we're really interested in looking at genetic modifiers that might explain why this occurs. And one hypothesis, for example, is that maybe there's a second genetic variant. And this one by itself doesn't cause disease, but when you add the 2 together, it can explain why one person with the same variant looks very different to another person. Maybe it's not just one variant, maybe there's multiple things going on. And if you recall, and I've talked about the human genome, we have, every one of us has roughly 3 million genetic variants. So it's probably a very complicated story and a lot of different factors playing together. And so this is very much, an active space of investigation and a very interesting area. And there's some interesting early sort of promising signs, but understanding, exactly what the role of all these other different genetic variants in explaining how someone's epilepsy will look is important. And if you think about, trying to go towards precision therapies, really understanding not just the single variant that causes disease, but a person's entire genetic background is potentially going to be very important and powerful for that. So I hope I've given you a little bit of a taste about what is bioinformatics. Maybe you never heard that word before, and what exactly we do. And so as I think is clear from this meeting, medical research is a massive team effort. Many people contribute in different ways. So, bioinformaticians working with clinicians, people in the lab, people doing animal studies, people trying to develop new drugs, genetic counselors. So we're really all working together to try and answer these important questions. So yeah, here's sort of a picture of the diagnostic odyssey, from the first symptom to then trying to identify a genetic cause to developing therapeutics. And hopefully you can see a little bit about what bioinformatics is and the role that we play in this incredibly important and big team endeavor. Thanks.